Okay, guys, so today we have lecture by digestive system. This topic you will have in after the module just next week, and this topic included in the next fifth module. So today we are going to start the digestive system because this topic is subdivided according to our practical lessons into 3 lessons. So today I'm going to give you information about the first 2 topics, and that will be useful for you for the next practical lessons. You might know from the previous information that digestive system main function is digestion. And from anatomy, you might know that digestive system consists of 2 parts. The digestive tract or alimentary tube, which includes all organs of the digestive system, starting from the oral cavity and up to the large intestine. And second component is associated glands. The associated glands, such as salivary, liver, and the pancreas, that produce the components for the digestion. Which components now we are going to talk in details in the question of the functional digestive system. So as I mentioned before, the main function is digestion. But what is digestion? Digestion is a complex process which involves the mechanical and chemical degradation of the food molecules, food particles. So the mechanical digestion is starting from the oral cavity. It's like chewing by the teeth and movements of the tongue, and also continuous in the next parts of the digestive or alimentary canal, such as peristaltic waves or peristaltic result of the muscular contractions of the digestive tube organs. And chemical digestion is a result of the influence of the digestive enzymes. So it is influence of the chemicals that provide splitting of the micromolecules to the monomers. Let's move. Guys, can you please switch off the camera and the microphone? All right, so we can see here general plan of the digestive tube structure. A digestive tube composed of the tunics that we are going to discuss right now, starting from the inner tunic or inner envelope. So we can see that the first tunic from the lumen is a tunica mucosa. Tunica mucosa composed of 3 components. First is lamina epithelialis, which is lined by different types of the epithelium depending on the organ of digestive tube. So, for example, in the oral cavity, it is lined by stratified squamous non-keratinized epithelium. In the stomach, small and large intestine, it is simple columnar epithelium. Second component of the tunica mucosa is lamina propria. Lamina propria is composed of the loose connective tissue, and this connective tissue reach for the blood vessels and the lymphatic capillaries. And the last component of the tunica mucosa is lamina muscularis. Over here we can see it. So the lamina muscularis is a smooth muscle tissue that have ability to contract. And next layer is tunica submucosa. So tunica submucosa, as we can see, is also loose connective tissue that contain blood vessels, nerves, as well as glands. But the important function of the tunica submucosa is that it forms a relief of the mucosa. What does it mean? If we will look on the lumen of the digestive tube, we will see the foldings or projections of the tunica mucosa. This is a result of the growth of the submucosa towards the mucosa. And these extensions or even invaginations are specific for the different parts of the digestive system. This is how we can distinguish the organ on the histological specimen by, first of all, the shape of its relief. So one more time, the submucosa form the specific relief of mucosa, and it's composed of the connective tissue. Also in submucosa, we can find the specific lymphatic aggregations that we call associated lymphoid, gut-associated lymphoid tissue. These aggregations of lymphoid tissue is important immunological barrier for digestive system organs that we will talk in details later. And the development of this lymphoid tissue also differ in the different organs. So now we are going to go to the next layer, next tunic. It's tunica muscularis. Tunica muscularis in different organs of digestive tract is represented by different types of the tissues. So anterior part of digestive tube, as you can see here, until upper third of esophagus, is represented by striated muscle tissue, striated or skeletal muscle tissue. The middle part, which is esophagus, small and large intestine, is represented by the smooth muscle tissue. However, posterior part is represented by the smooth and striated muscle tissue. So how to memorize which type of tissue lines each segment? We need to remember for that what is the type of contraction for the skeletal and the smooth muscle tissue. Who can tell me what is the type of contraction for the skeletal tissue? Does skeletal muscle tissue contract voluntary or involuntary?
Voluntary.
Voluntary.
Yes. Correct. Yes, because it is rated by a somatic nervous system. Yeah, thus we can control it by our will. So thus the upper third of esophagus, the oral cavity, the pharynx, and the upper third of esophagus is lined by striated muscle tissue, because we know that swallowing, or a speech is a voluntary process. That is why those organs are lined by or represented by the skeletal muscle tissue. However, we cannot control the peristaltic waves of our small intestine or secretion of our stomach, and this is why these organs are lined by the smooth muscle tissue. The posterior part, just the distal part of the rectum, is represented by both types of the tissues. So that was tunica muscularis, and the last tunic is tunica serosa. So external tunic can be represented by serosa or adventitia. Let's see the differences between them. So adventitia, tunica adventitia, is loose connective tissue. Tunica serosa is loose connective tissue covered by the simple squamous epithelium that is called mesothelium. So now let's see which organs are lined by the adventitia and which are lined by serosa. The anterior part above the diaphragm and posterior part is aligned by tunica adventitia. The organs that are of the middle part, that are covered by sorry, that are covered by peritoneum. The peritoneum is a serous membrane, and this forms this simple squamous epithelium or mesothelium from outside. So the middle part of the digestive tube, the 2/3 of esophagus, small large intestine, lined by tunica serosa. Okay, one more time, guys. So that was general plan of digestive tube or the alimentary canal. That means that all organs, esophagus, stomach, small and large intestine, are represented by these four tunics. But as you might mentioned before, different organs have their structural features. And now we are going to focus on the structural features only, but you need to keep it in your mind, the general plan of the structure. Yeah, the tunics and the layers of each tunic. Yeah, tunica mucosa, tunica submucosa, tunica muscularis, and tunica externa. So please remember it because it will help you to describe each organ when when you will discuss this topic on the practical lessons with your teachers. So we are going to start now with the oral cavity, and the organ that we need to focus on is the tongue. The tongue is a muscular organ that provide many functions in our body. First of all, yeah, it's an organ of it's a taste organ. This organ contain taste receptors that are important, that provide receptor function. Second, it is responsible for the process of the speaking, and also it helps in the movement of the food, in the process of the swallowing of the food. So let's go to its histological structure. And in this spacement, we can see that the tongue composed of two surfaces. The superior surface that's sometimes called dorsal surface, and inferior surface. Okay, guys, I'm sorry, this here it's mistake in my slide. I will change it out after the lecture and send you PDF without mistake. So the superior surface of the tongue consists of such components of tunica mucosa as stratified squamous, parakeratinized epithelium, and lamina propria. Let's focus in details on each component. So the superior or dorsal surface of the tongue, first of all, has great developed papillae that we will discuss later. So this is first diagnostic criterion, how you can distinguish it, and also the difference in the layers. So the superior surface has only two components: stratified squamous, parakeratinized epithelium. What does it mean? Stratified, you might remember that this epithelium composed of many layers, yeah, more than one layer of the cells. Parakeratinized, remember from the previous semesters that in case of stratified epithelium, we have keratinized and non-keratinized epithelium. For example, our skin represented by keratinized, stratified squamous keratinized epithelium. However, for example, if you remember from the eye topic, the cornea is lined by non-keratinized epithelium. And the main difference between them in the stratum corneum. So the stratum corneum in non-keratinized epithelium is absent. In case of the dorsal surface of the tongue, some areas of the papillae contain have process of keratinization, means the death of the keratinocytes. Some areas have no keratinization, and due to this, we call this epithelium parakeratinized, means it's intermediate by its properties between the keratinized and non-keratinized epithelium. Lamina propria is underlying the epithelium. It's underlying the epithelium. So the lamina propria is a loose connective tissue that contain blood vessels and the nerves for the blood supply for the nourishment of the epithelium. After this, you can see the numerous skeletal muscle tissue fibers, bundles of the fibers, because as we mentioned before, the tongue is a muscular organ. The inferior, so here is the structure of inferior surface. Sorry guys, this is mistake, not superior, inferior surface of the tongue is represented by two components. So the tunica mucosa and tunica submucosa. So as you might see from this, if we will compare these two surfaces, you might mention that the inferior surface of the tongue has tunica submucosa, and however superior surface has no submucosa. So the inferior surface of the tongue composed of stratified squamous non-keratinized epithelium, over here we can see it, and lamina propria, which is loose connective tissue, and tunica submucosa. The tunica submucosa has great developed blood supply, and because of this we prescribe some medicals sublingually, because the contaminants or of the molecules of the chemicals just by diffusion reach the blood supply from the oral cavity. Yes, so it's much more quicker comparing to the peroral process of the absorption of the nutrients. So let's summarize, guys. So the main difference between the superior and inferior surfaces that of the tongue, that the inferior surface, except tunica mucosa, has great developed tunica submucosa with blood vessels and the nerve plexuses. And also the second difference in relief, the superior surface of the tongue has great developed papillae, which are long extensions. And now we will focus in details on the structure of the tongue papillae. So the papillae are projections of the epithelium, as you can see, and lamina propria, and we differentiate such types of papillae as fungiform, foliate, circumvallate, and also these filiform. So the fungiform, foliate, and circumvallate papillae contain test buds. Over here we can see the lateral surface of papillae, and we can see that each of these papillae, fungiform, foliate, and circumvallate, contain the taste buds. In this diagram, we can see the cellular composition of the taste bud. The taste bud consists of three types of the cells. The gustatory cells, over here we can see them as a bright violet color. The light pink color are supporting cells, and a bright pink color in the base are basal cells. So each of these cells provide their own role. The gustatory cells are basically receptors. They are chemoreceptors that accept the dissolved in saliva chemicals that sent to the brain about the quality of the food that were eaten. The supportive cells that are situated between the gustatory cells provide support of them, and also basal cells that are responsible for the process of the regeneration. So in the apical surface of the taste bud, we can find the taste pore. The microvilli of gustatory cells are found in the taste pore. Thus, they act as the sensory parts of the cells that accept the stimuli, which are chemical molecules, and lead to excitation of the sensory hair cells that in their base interact with the afferent fibers that bring information to the brain about their food. So one more time, this process is important for the checking the quality of the food that we add, and thus provides a protective role. Yes, so the taste sensation is important function, and one more time, it is provided with the help of gustatory cells in the taste bud. We move on, and next component that we are going to discuss are salivary glands. So the salivary glands, according to classification, we subdivide into the large and small glands. So the large glands are parotid, submandibular, and sublingual. The small glands we can find in the tunica mucosa of the lips, tongue, palate, cheeks. For example, if we will return back to the previous diagram, over here in the tongue we can see the small salivary gland, the secretory portion. So the salivary gland, being the typical exocrine gland, consists of two parts: the secretory portion and the duct system. And the ducts of the salivary glands open to the oral cavity. For example, over here we can see the duct of the parotid gland. So the large salivary glands have a typical compound tubular alveolar glands. What does it mean? Compound means that these ducts have branched, these glands have branched duct system. Tubular alveolar means that in the secretory portion of these ducts there are sac-like acini and tubular acini. According to the cells type of secretory portion, we subdivide three types of secretory portion. But now let's firstly focus on the cells. So each secretory portion, we differentiate two types of the secretory cells in secretory portion. The serous cells, which are protein secreting cells, which have a basophilic cytoplasm, and these cells are usually not large, so they are small. And the mucous cells. The mucous cells are much bigger and they have light cytoplasm. The color of the cytoplasm is a result of the secretory products of each of the following cells. So now let's focus on the types of the secretory portions or acini. We differentiate three types of the secretory portion. The serous acini, over here we can see it in the top diagram. The serous acini composed of the serous cells. The serous cells, if you remember, are responsible for the production of the proteins. This is why in their cytoplasm abundance of the rough endoplasmic reticulum gives basophilia to the cytoplasm. So this is why these cells have bright color of the cytoplasm. Mucous acini, which composed of the mucous cells only. So in case of the mucous cells, we mentioned that these cells are responsible for the production of the mucin, which is packed in the mucinogen granules, and this is why the cytoplasm has light color or pale color. And mixed acini, the mixed acini composed of both types of the cells. In the center, we can find mucous cells with the light cytoplasm, and in the periphery, the mucous cells surrounded by the serous cells. These serous cells form caps, which called demillions of Giannuzzi. So these mixed acini, one more time, composed of both types of the cells, the mucus in the center and the serous on the periphery. Ducts. As I mentioned before, each salivary gland composed of the secretory portions and the ducts. And we differentiate four major types of the ducts. First type is called intercalated duct. The intercalated ducts we can find near the secretory portion. Over here we can see them. So the intercalated ducts are the smallest, and they are lined by simple cuboidal epithelium. In the zoom in, we can see this typical structure of intercalated duct. Second type is intralobular. Intralobular, intra prefix means inside. Inside the lobule. Over here we can see connective tissue stroma that separates the parenchyma of the gland into the lobules. And inside each lobule we can see the intercalated and intralobular ducts. So intralobular ducts, or its second name striated, lined by simple columnar epithelium. Over here we can see this cylindrical shape of the cells. These ducts are bigger comparing to intercalated ducts. Yeah, so here we can see intercalated duct and here intralobular. So these cells, the cells of the intralobular duct, are characterized by the basal striations. Now we will see in details the nature of the basal striation, and due to this, these ducts are called striated ducts. So basically, basal striation is enfolding of the plasma membrane, and in the base of these cells we can find mitochondria arranged parallel to the folded membrane. And the main function of the striated duct is reabsorption of the sodium ions. This is why the salivary salivary glands maintain their sodium homeostasis, and also it keeps a pH of saliva. So let's see what is the nature of striated duct. Over here in this diagram, we can see the intercalated duct. So this is secretory portion. This diagram, guys, by the way, you will need to draw in your albums, so it will be useful. You can find it in Jankira textbook. So here we can see the acini. We can see the secretory portion, the serous acini, the mucous acini in the cross section, intercalated duct, which is lined by simple cuboidal epithelium, and striated duct, which is lined by simple columnar epithelium. And as you can see in the base of each cell, we can find the basal striation. The basal striation is elongated mitochondria that are found in the basal surface of the cell. Why mitochondria? Because we know that sodium transport requires energy of ATP. Yes, so it's active process. This is why the mitochondria produce energy support for the process of the reabsorption of the sodium ions in intercalated ducts. In the intralobular ducts. Next duct is intralobular. Over here we can see the intralobular duct. The interlobular. Yeah, so let's differentiate. Intercalated in intralobular duct we can find inside the lobule. Between two lobes we can find inter. Inter prefix means between. Between two lobes we can find interlobular or excretory duct. So interlobular or excretory duct is lined by stratified cuboidal or columnar epithelium. Over here we can see two layers of the cells. Yeah, this is what it make it different from the intralobular or intercalated duct. So interlobular is the biggest duct, and it's open to the duct or to the oral cavity, and this duct once again we can find in between the lobes in connective tissue stroma. So here we can see this connective tissue between two lobes, and inside it we can see this large diameter interlobular duct. Now we go to the salivary glands, to the features of each gland. Large salivary glands, if you remember, are parotid, submandibular, and sublingual. So we are going to start with the parotid gland. What are the structural features? How we can differentiate this duct or this gland on the specimen? First of all, the parotid gland is represented by the serous acini only. So if we will look in the specimen, we can see only serous cells. Yeah, and this is why we can. This is the only one gland that has typical serous that composed of the serous acini only. These serous cells are responsible for production enzyme alpha amylase. The alpha amylase enzyme are responsible for the splitting of the carbohydrates, and also so we can say that thus the parotid gland initiate the chemical degradation of the carbohydrates in the oral cavity, because the final or terminal degradation will happen in the small intestine of the carbohydrates. So one more time, guys, parotid gland is the only one gland that consists of the serous acini only. Now we can compare parotid gland with submandibular and sublingual gland. Submandibular gland, as we can see in this diagram. Can you please switch off your microphone? Thank you. Okay, submandibular gland has two types of the serous of the acini. The mixed acini, yeah, so we can see over here typical mixed acini. The mucous cells in the center and the serous cells on the periphery, which we can see as the demillions of Giannuzzi. So this mixed acini, and here we can see the typical serous acini. So the main secretory products of the submandibular glands are alpha amylase, which we mentioned before. Yeah, so this enzyme degrade the carbohydrates in the oral cavity, and also lysozymes that provide have antibacterial properties and split the bacterial wall. Next, sublingual gland. In sublingual gland, we can see predomination of the mucous acini. So we can see numerous mucous cells, and we can see also somewhere we can see also mixed acini, but mainly mucous acini. So these glands mainly produce an alpha amylase and also lysozyme. So guys, one more time, these three glands have different types of the acini. In the parotid gland, typical serous acini, and submandibular mixed and serous acini. In sublingual, mainly the mucous acini. Okay, what else we did not mention is about so-called myoepithelial cells. If you will look on this, on the cross section of this diagram, in this diagram you can see the cross section of the secretory portion. Myoepithelial cells surround the secretory portions, and they have. Guys, please don't switch on your microphones. So the myoepithelial cells have the myofilaments in their cytoplasm. So the myoepithelial cells have the microfilaments in their cytoplasm that are able to provide contraction and stimulate secretion of the secretory cells in the secretory portion. So thus myoepithelial cells, we can say, stimulate release of the salivary components. Now let's move on and let's discuss the esophagus. Esophagus is the elongated tube, and this organ over here we can see in the cross section. Yeah, this organ lumen changes as a result of the transport of the chyme or the undigested food through the esophagus. So let's talk about the structural features of esophagus. First of all, we need to focus on the tunica mucosa. So the tunica mucosa of esophagus is lined by the stratified squamous non-keratinized epithelium. Over here in zoom in, we can see this type of epithelium. Yeah, we can see many layers of the cells, and the stratum corneum is absent. So this is first structural feature. Second is dedicated to relief of esophagus. If you remember, in the beginning of the lecture, I told you that tunica submucosa, which grows towards the mucosa, forms specific shape of the surface. And this shape is different in different organs of the digestive system. So we can see in the cross section of esophagus that esophageal mucosa is contain the folds, longitudinal folds. Yeah, here we can see them. These folds will be extend in response to the chyme passage. Also, if we will look into lamina propria of the tunica mucosa, we can see in the cardiac region of the esophagus, which is distal part where the esophagus are bordered with the stomach. So in the distal part of the esophagus, we can find the cardiac glands. Only in this part of the esophagus, we can find the cardiac glands. The proper esophageal glands are found in the submucosa. So here we can see tunica mucosa, here is submucosa, yeah, and here we can see the proper esophageal glands in tunica submucosa. Muscularis of this esophagus has two layers, and outer and inner, and these layers are represented by different types of the tissue according to their part of esophagus. If we subdivide esophagus into three parts, the upper third of esophagus is represented by the striated muscle tissue. And you remember from the beginning of the lecture, because this part is situated in the border with the pharynx, and thus it has the same type of the muscle tissue on the upper third of the upper part of the digestive tube. The middle third of esophagus is represented by both types of the tissue. Over here you can see the smooth and the striated muscle tissue, both are found in the middle third of esophagus. And the lower third of esophagus is represented by the smooth muscle tissue only. So this is a second structural feature. And also we remember that esophagus, it's an important organ for the which communicates the oral cavity with the stomach. So the stomach, as you can see, is a sac-like organ that provide many functions. First of all, it stores the food that coming from the esophagus. Also, it provides the chemical digestion of the food, because the stomach, the cells of the stomach produce digestive enzymes such as pepsin and gastric lipase that provide initial digestion of the proteins and the lipids. Except this, the stomach produces intrinsic factor. The intrinsic factor is an important component for vitamin B12 absorption in the small intestine. And this vitamin, if you remember, plays important role for the maturation of the red blood cells in the red bone marrow. Also, absorption of the water, salts, sugars, alcohol, and endocrine secretion. Now we will focus on the structure of the stomach. And if you remember from anatomy, anatomically, stomach is subdivided into four regions: the cardiac region, the fundus of the stomach, the body, and the pylorus. So in case of histological structure, we subdivide stomach into three parts: the cardiac, fundus together with the body called fundic, and pyloric. So this classification is a result of the different types of the glands. So let's focus on the structural features of the stomach. We can see that first of all, the features of relief. If you remember, in the esophagus, it was longitudinal folds. In case of the stomach, we also can find the same longitudinal folds. Except these, we can find gastric pits or alveoli. It's invaginations of the epithelium into lamina propria, and at the same time, these invaginations plays a role of the ducts of the gastric glands. In the end, in the base of each gastric pit, we can find gastric gland and mammaliated area. So one more time, the specific relief of the stomach that we can find only in this organ are gastric pits and mammaliated areas. So over here in the spacement, we can see gastric pits in the top, and in the base, we can see gastric glands as a secretory portion of the gastric glands. So let's move on. Second structural feature, epithelium. It's a simple columnar epithelium. Remember, in esophagus it was stratified squamous, non-keratinized. So in the stomach, as well as the small and the large intestine, epithelium is simple columnar. But because in the stomach almost all cells have secretory activity. We call this epithelium secreting. So lamina propria, second structural feature. Lamina propria contain gastric glands. Remember, in esophagus, we could find typical esophageal glands in tunica submucosa. In case of the stomach, over here we can see mucosa, tunica mucosa, and we can find, so this is epithelium. Over here we can see the gastric glands. So the gastric glands, as I told you before, according to the region of the stomach, we differ into the fundic glands in the fundic region, or gastric glands in the fundic region, cardiac glands in the cardiac region, and pyloric glands in the pyloric region. So remember, we talked about, when we were talking about this esophagus, we said that in the door on the posterior surface, in the lower third of esophagus, we can find cardiac glands. So this is result of the connection of the esophagus with the stomach. Yeah, the regions that are situated close to each other have similar histological structure. So now we're going to focus on the structure of the gastric glands in details. So each gland has 3 segments: the short isthmus, the neck part, and the base or fundus. Now we need to focus on the cellular composition of the gastric glands. We differentiate 5 types of the cells. The chief cells, over here we can see these cells in the base of the gland with the basophilic cytoplasm. Parietal cells also found in the base of the gland, but in contrast to the chief cells that form the groups of the cells, the parietal cells are isolated, so they do not form clusters. Mucous neck cells found in the neck region of the gland. They have light cytoplasm because of the mucogen granules. Undifferentiated stem cells also found in the neck part of the gland. These cells are responsible for regeneration. This is why they have small size. And enteroendocrine cells, these cells have endocrine activity, provide endocrine activity, and produce different biological active substances and hormones that release to the blood. We can find these cells in the base of the gland. Now let's focus on details on each cell, and let's start with the chief cell. So the chief cells have bright basophilic cytoplasm and are found in the base of the glands. So the chief cells produce the enzymes, the pepsinogen and gastric lipase. The pepsinogen is in active form and in the lumen of the stomach by the influence of the gastric juice. It is converted to the active form pepsin. The gastric lipase digest the lipids, so it provide initial digestion of the lipids in the stomach. So the basophilia of the chief cells is a result of the abundance of the rough endoplasmic reticulum in the base of the cell, and this rough endoplasmic reticulum produce the components of the enzymes, the pepsin and the gastric lipase, because enzymes, if you remember, have the protein nature. So how we can distinguish the chief cells among other cells? First of all, by their basophilic cytoplasm, and second, because they are found by their localization in the base of the gland, and also that these cells are the most numerous. They form the groups situated close to each other, And these cells, as we mentioned before, have a secretory activity. Other cells, also important in the stomach, called parietal cells. These cells produce hydrochloric acid. Hydrochloric acid, which provide low pH of the gastric juice, and intrinsic factor. So let's focus first of all on the role of the hydrochloric acid. So the hydrochloric acid, if you remember from the previous slide, is an important component for the activation of the pepsinogen. When pepsinogen released to the lumen in response to the hydrochloric acid, it is converted to pepsin. Intrinsic factor is the molecule that's important for the absorption of the vitamin B12 in the small intestine. And as I mentioned before, it is the vitamin B12 is important component for the maturation of the red blood cells. Now let's focus on the structure of the parietal cells. In the cytoplasm of the parietal cells, we can see numerous mitochondria, which give oxyphilia to the cytoplasm. Also, we can distinguish these cells by intracellular canaliculi. These intracellular canaliculi take part in the transport of the ions. We mentioned before that the parietal cells secretes hydrochloric acid. So the hydrochloric acid composed of two ions, hydrogen and chloride. Yeah, thus the transport of these ions they take place in the intracellular canaliculus, and with the help of the proton pumps, it releases on the surface of the cells where it binds with the chloride ions and become the hydrochloric acid. Mucous neck cells. The mucous neck cells are found in the surface of the gland in the neck region, and these cells secrete mucus. The role of the mucus is, first of all, protective role. The protection against the aggressive low pH of the stomach. We know that pH of the stomach is from 1 to 3, and this pH provides protective role against microorganisms. So if there is any pathogenic flora invades our body, a digestive system with the food, the hydrochloric acid splits the bacterial wall, and as a result, it provides protection. But at the same time, it may damage this tunica mucosa of the stomach. To prevent this process, to prevent this chemical burning of the mucosa, the mucous neck cells secretes the mucus that lines the surface of the epithelium of the stomach, and as a result inactivate or neutralize hydrochloric acid on the surface of the stomach and protects its wall. Enteroendocrine cells, next cell that have endocrine functions. So if you remember, when we were talking about endocrine system, we mentioned about diffuse neuroendocrine cells. So these cells do not form glands, the endocrine glands, but they are found as isolated cells in the tunica mucosa of many internal organs, especially abundance of these cells are found in the digestive system. In the stomach, these cells produce such substances as gastrin, glucagon, serotonin, substance P, VIP, and these cells, if you remember from endocrine lecture, provide paracrine regulation. What does it mean? They regulate neighboring cells and stimulate secretion of the secretory products of these cells. So these cells provide important role for the regulation of the organ where they are found. These cells also found in the base of the glands, and thus how we can differentiate. Let's go back to the previous diagram. Yeah, over here. How we can distinguish the endocrine cells, the chief cells, and the parietal cells. How we can differentiate these three types of the cells. First of all, yeah, because all of them are found in the base. In case of the chief cells, they are also secretory cells, but these cells release their secretory products, which are enzymes, into the lumen of the stomach. Yes, so the pepsinogen and gastric lipase, after secretion, accumulated in the apical part of the cell in the secretory granules. So after this, these secretory products released by exocytosis via the apical surface of the cells. However, the G cells accumulate their secretory products, which are hormones, in the base of the cells, because the endocrine cells released their secretes or hormone into the blood stream. Yes, so over here we can see the accumulated secretory products, secretory vesicles, sorry, in the base of the cell. The same on electronomytophotograph that we can see in the specimen below. So we can see these black dots as the secretory granules of the enteroendocrine cells that contain numerous hormones, and as we can see here, they release their secrets to the blood capillaries. Over here we can see the fenestrated blood capillaries, which absorbs these hormones. Also, the next feature of the stomach is the tunica muscularis. Remember, in esophagus we mentioned about 2 layers of the tunica muscularis. In stomach there are 3 layers: the inner oblique, middle circular, and outer longitudinal. And we can find 2 nervous plexuses in the stomach. The first, submucosal or Meissner plexus, is found just under the subtunica submucosa, between the submucosa and muscularis. Mesenteric or Urbach plexus is present between, over here we can see it between the bundles, between the layers of the tunica muscularis. Both first of all regulate the blood supply in the stomach, and also stimulate contractions of the stomach wall, and as a result they stimulate peristalsis of the movement of the chyme through the digestive tube. Small intestine, the next organ that we need to discuss, and here you can see that the small intestine anatomically is subdivided into the 3 segments: duodenum, jejunum, and ileum. So the functions of the small intestine is terminal digestion of the food, because we remember that initial digestion of the carbohydrates happened in the oral cavity. Initial digestion of the proteins and lipids happened in the stomach, and the terminal digestion of the food happened in the small intestine. This digestion, chemical degradation of the food in the stomach, in the small intestine, happened with the help of the digestive enzymes of the pancreas. Also, nutrient reabsorption, so the absorption of the monomers from the lumen of the small intestine. So if we will look into this diagram and the spacement, we can see the specific relief of the small intestine. So first feature of the small intestine that will help you to distinguish it from the stomach is its specific relief. We can see that the small intestine form the, first of all, it has the plica circularis or folds. Also in zoom in over here, we can see the villi, which are projections of the lamina propria into epithelium, and crypts. Crypts are invaginations of the epithelium into lamina propria. So one more time, finger-like projections we call villi, invaginations of the epithelium we call crypts or crypts of the lubricule, or sometimes you can find it as interstitial glands. So this specific relief we can find only in the small intestine. Remember, in the stomach it was the gastric pits. Here we can find villi and interstitial crypts. Yeah. For example, in the large intestine, there are we will distinguish interstitial crypts, but villi are absent in the large intestine. This is why villi are the typical for the small intestine only. Let's focus on the epithelium of the small intestine. So the cells of the small intestine are enterocytes, and these cells we can find. Now we will talk about common cellular composition of the crypts and villi. So enterocytes, the most numerous cells, we can find them. I'm sorry. We can find them in both cases, in villi and crypts. Goblet cells are less present, but they are still abundant, and we can find them in also in case of the villi and crypts. These cells have light cytoplasm and produce mucus. Next cell is Paneth cell. Paneth cells we can find only in the crypts of the small intestine. Enteroendocrine cells. The enteroendocrine cells usually also found in the crypts. Microfold cells and stem or undifferentiated cells. Let's focus on the cellular, each cell in details. But before we will start, let's remember what is the main function of the small intestine. The terminal digestion and absorption of the monomers to the blood. Thus, this function provide the most numerous cells, the enterocytes. So the enterocytes absorb the nutrients, which they after this pass to the blood capillaries, which are found
Excuse me, ma'am, your voice is not audible. Ma'am, your microphone is mute.
I see. Yes, thank you so much. When did my voice was stopped? When you did not? When you,
Just few seconds ago, it was
just
not.
few seconds. Okay. Yeah. So I can continue. Yeah. Thank you so much, guys. Inform me next time if it will happen, please. Okay. So one more time, monomers, glucose, and amino acids are absorbed into the blood capillaries. The lipids like fatty acids and glycerol are absorbed in the lymphatic capillaries and then drained to lymphatic vessels. So one more. Now we will focus into the cellular composition of the villi and crypts, and we will start with enterocytes. Here you can see the light microscopy of the enterocyte and the scan electron-microphotograph. Guys, can you please switch off your microphones? So the enterocytes, as we mentioned before, the most numerous cells. I'm sorry, guys. There's some. Okay. So the most numerous cells. How we can distinguish these cells? These, remember we said that in the stomach we have simple columnar epithelium. So these cells are tall columnar, and in the apical surface of each enterocyte we can see the brush border. What is over here, especially in the scanned electron microphotograph, it is visible. So what is brush border? It is microvilli that increase the surface area for the process of absorption. So this is important for the transport of the nutrients from the lumen to the blood capillaries. So one more time, enterocytes are the typical and the most numerous cells of the small intestine that are responsible for the process of the reabsorption. Next are goblet cells. So the goblet cells got their name due to their shape. Yeah, this typical champagne glass. And we can see on the apical surface of the cell mucogen granules. The main function of the goblet cells is the production of the mucin. Still some microphone on. Okay, so mucogen granules that produce mucus. So what is the function of the mucus in the small intestine? First of all, it's lubrication. Yeah, to provide, it lubricates the lining of the small intestine for the transport of the chyme, and also it protects the tunica mucosa against influence of the hydrolytic enzymes that released from the pancreas for the chemical digestion of this on the smallest intestine. Pennant cells. The pennant cells, one more time, are found just in the base of the crypts. So over here we can see invagination of the small intestine, which we call crypt of the luberculum, and over here we can see the pennant cells. The pennant cells we can distinguish by their polarity. So these cells, sorry guys, there is some. So these cells have bright acidophilic apical surface and bright basophilic base. So this polarity is a result of the secretory function of this cell. So these cells, the pennant cells, produce enzymes, antibacterial enzymes, such as lysozyme and other enzymes that are enzymes that provide digestion of the bacterial wall. So these enzymes are packed into their secretory vesicles, and this we can see in the which we can see in the apical surface of the cell. The basal surface is bright basophilic because of abundance of the rough endoplasmic reticulum. So one more time, the pennant cells are secretory cells that are found in the base of the interstitial crypts and responsible for production of the hydrolytic enzymes that split bacterial wall and provide protective role. Enteroendocrine cells of the small intestine are typical diffuse neuroendocrine system cells that produce such hormones as cholecystokinin and secretin. So these two hormones affect secretion of the pancreatic cells. So thus we can say, yeah, that they regulate secretion of the pancreatic enzymes, and as a result, these provide important role for the digestion of the chyme in the small intestine. Another cells that we need to talk about is microfold cells. The microfold cells are overlie the gut-associated lymphoid tissue in the small intestine, and these cells, these microfold cells, provide transport of their antigens, and as a result, they provide protective role as well as the immunocompetent cells. Stem cells or undifferentiated cells are we call so because they are they have ability to divide, and as a result they provide regeneration of the epithelium of the small intestine, of the crypts and villi epithelium. Let's watch the video now together about the microfold cells. I will. You can find this video in our website. So if there will be any problems with the watching, you can do it later. So we can see the surface. We can see the relief of the small intestine. We can see numerous villi that provide the process of the absorption of the nutrients to the blood. Now we will focus on the cellular composition, and we will check the role, we will see the role of the M cells in the small intestine. So now we can see the digestive system structure, and we can see that if what happened when some food and probably some pathogenic flora invades our digestive system. Yeah, it passes through esophagus, then go to the stomach, and if it was not eliminated in the stomach, then in the small intestine, these microorganisms will be eliminated with the help of our immune system cells. So let's see what happened exactly. Over here we can see M cell. So we mentioned before that the M cell overlies the lymphoid cells and lymphoid system cells, and here we can see these lymphatic aggregations that we call the component of the gut-associated lymphoid tissue. So we can see the villi and lymphoid tissue. Together they are connected because they both drain lymph, and thus they bring it to the lymphatic nodules that contain a great number of the lymphoid, the great number of the lymphocytes. Over here we can see the enterocyte and M cell. Yeah, so the M cell, as we mentioned before, provide the transport function for any pathogens, and as a result help to immune cells to recognize and eliminate them. So, for example, if some bacteria invade our body, the M cell passes to the macrophage cell, and if you remember, the macrophage or dendritic cell, they both antigen presenting cells. So here we can see that the the dendritic cells pass its long process that also phagocyte these bacteria, and after this go to our lymphocytes, immunocompetent cells, and activate the immune response, T and B cells. So yeah, it represents information for immunocompetent cells, and thus these cells start to initiate immune response. Okay, later I will show you the video about them. Later I will show you the video about the mechanism of the regeneration. Now just let's move on and let's talk about the structural features of the different parts of the small intestine. Remember that small intestine composed of these three segments: the duodenum, jejunum, and ileum. So how we can differentiate? Sorry? Is there any problem, guys? No? Okay, we continue then. Okay. So, one more time. How we can differentiate the duodenum from jejunum or ileum? First of all, by the relief. All these three segments contain, if you remember, the villi, cretes, and what is the difference between the duodenum and jejunum? The duodenum is in initial part of the small intestine, and the villi there are shorter and more wide. However, in the jejunum, the villi are more taller and more thin. Second difference, that in duodenum we can find so-called duodenal or Brunner glands in the submucosa. So these duodenal or in submucosa and mucosa. So these duodenal or Brunner glands also produce enzymes for the additional digestion on the surface of the villi and cretes. Second structural feature is dedicated to the ileum. The ileum contains great developed mucosa-associated lymphoid tissue that are called pear pouches. The pear pouches over here we can see in the small intestine are aggregations of the lymphoid tissue in tunica submucosa, which, as we mentioned before, underlying the M cells. So that all parts of the small intestine contain gut-associated lymphoid tissue, but its volume increases toward to the distal parts. That is why it's great. The ileum get its great development as a pear pouch. Large intestine. The large intestine is the last segment of the digestive system. The large intestine function is the terminal digestion of some components that were not digested in the small intestine, for example, large carbohydrates. Also, it provides absorption of vitamin K and also the large intestine contain the bacterial flora. These microorganisms provide a protective role against pathogenic flora in case of its invasion to the digestive system. So what are the structural features of the large intestine? First of all, it's about its epithelium. So epithelium of the small intestine contain a great number of the goblet cells. Remember, in the small intestine, we said the most numerous cells are enterocytes. because the main function of the small intestine is absorption of the nutrients. However, large intestine is a terminal part of the digestive system. Also, if we will look into relief of the large intestine, we cannot see the finger-like projections, which we call villi, only invaginations or crypts. Yeah. So here we can see the numerous crypts of the large intestine. So this is a second structural feature. Also, the muscular layer of the large intestine consists of the tunica muscularis of the large intestine consists of two layers as well as the small intestine, the inner and outer layer. However, the outer layer is discontinuous and it forms the so-called tinea coli longitudinal bands. Over here we can see it on the specimens. These longitudinal bands, the contraction of these longitudinal bands stimulate peristaltic waves in the small intestine, and as a result, the transport of the chyme through the large intestine. So, guys, the last question that we need to talk is embryonic development of the digestive system. If you remember from the embryology, the endoderm is a main source of the digestive and respiratory system. However, the invagination of the oral ectoderm give rise to the components of the oral cavity and basically which is anterior part of the digestive tube and posterior part of the digestive tube. Thus, the epithelium of these organs of the anal and oral cavity are lined by the ectoderm, the epithelium as derived from ectoderm, and other parts of the digestive tube, the middle part of the digestive tube, as well as digestive glands, are lined by the endodermal origin epithelium. So guys, this is shortly about the digestive system. Now, if you have any questions, please feel free to ask me. At the end, I would like to show
Ma'am, your mic has been muted.
Thank you so much again. Somebody do it again. I don't know why. Okay, guys. Before we will finish, I want to show you the video about the process of the regeneration of the epithelium of the small intestine with the help of the stem cells. Yeah. So, if you have any questions, please feel free to ask me now. Okay, so now I'm going to demonstrate my screen and show you the video. So now we go to the small intestine. Remember, the crypts of the small intestine contain great number of the stem or undifferentiated cells. So these cells provide regeneration of the epithelium, and in case of any damages, in case of the chemical changes of the other cells, the stem cells are able to replace the damaged area because these cells are able to divide. So due to mitotic activity of these cells, we have regeneration of the epithelium. Over here we can see the villi or base of the intestinal glands. We can see the pennant cells and the stem cells in the base of the crypt. And over here we can see the process of mitosis. Mitosis which is important for the regeneration. And as you can see, these new formed cells will be able to differentiate in any types of the cells, in goblet cells, in enterocytes, and as a result, they provide a regeneration of the epithelium of the crypts and villi both. But at the same time, in case of uncontrolled process of mitosis, these cells, yeah, and you can see how the cells die in the top of the villi in response to the influence of the unharmful some harmful substances. So at the same time, rapid mitosis or uncontrolled mitosis which is not regulated by our immune system, may lead to formation of the adenomas or cancer transformation of the secretory cells of the small and large intestine. So now we will see the pathological mitosis, which may lead to the formation of adenoma. So we know that our immune system have protective mechanisms that control the formation, that prevent formation of the cancer processes. But at the same time, in case of any alterations, it may lead to formation of the abnormal cells, which has modified DNA, and these cells have rapid mitosis because their mitotic division is not controlled by immunocompetent cells, and as a result, they may lead to formation of the adenoma of the digestive system. Okay, guys, this is clinical correlations. One more time, for the next week, we need to study digestive system starting from the oral cavity up to esophagus, including esophagus. So in the oral cavity, I did not mention about the structure of the cheeks, lips, soft and hard palate, but these organs are given in Jankira textbook. So the tongue, salivary glands, and esophagus I already represent you today. So next week, one more time, you will have accept the discussion or oral discussion of the topic with your teachers. You will have a Moodle test. So those students who had any problems with the Moodle forms during the seminar lessons, please try to contact Dean office and ask and find solution to sort out these problems, because we will use Moodle tests every week till during this semester, and you will need to write the Moodle MCQ on each practical lesson as well as seminar lessons. Okay, guys, do you have any questions from now on?
No, ma'am.
Okay, guys, then if you have no questions, I will send you over here to the chat, to the Skype chat, the PDF of my lecture later. And after this, we will download this PDF on the website as well. Just if you will, after some time, maybe on weekend, so you can watch it anytime and get yourself ready for the next week. Okay, guys, if you have no question, I appreciate you for your presence, for participation in discussion, and I wish you good luck, those who still do not have module in their module. And we will meet in 2 weeks in the next lecture. So next lecture we will also have in the Skype. You can find the hyperlink to the Skype in our website, and join it before the lecture. Okay, guys, then for today we finish. Thank you for your attendance and see you next lecture. Goodbye.
Thank you so much.
Bye, ma'am. Goodbye, teacher.
Bye.